Good afternoon, everyone. Today, I would like to talk about association of genetic polymorphisms with complications of implanted LVAD devices in heart failure patients. Left ventricular assist device is one of the alternative treatments for heart failure patients instead of heart transplantation, which improves patients' quality of life and reduces mortality rate. However, in the first year after LVAD implantations, patients have complications such as thrombosis and bleeding events. To reduce thrombosis events, patients usually prescribed with anticoagulation treatments such as warfarin and aspirin. But due to the incorrect dosage, patients still have thrombosis and the bleeding complications. Another reason of the development of the complications is shear stress at the blade region of the LVAD's rotary, which causes platelet dysfunction. Apart from the platelet dysfunction and the anticoagulation treatment, the incorrect one, the inherent genetic information of patients may also have increased risk to the development of the complications. Consequently, the aim of our research is to identify influence of the polymorphisms encoding the coagulation system of the process of the hemostasis, metabolism of the anticoagulant treatment, which impacts to the development of the complications in heart failure patients. Our research included heart failure patients with implanted LVADs and a control group, which are healthy group, for comparison. Heart failure patients were divided into two groups for comparative analysis, such as with complications and without complications. DNA samples were genotyped for 21 SMP polymorphisms. Our investigations identified 5 polymorphisms in 3 genes which are significantly associated with heart failure patients' complications. 2 of them were in the VCORC1 gene, which is responsible for the metabolism and the action of the warfarin. The VCORC1 gene helps to identify correct dosage of the warfarin treatment for patients. Another polymorphism was in ITGP3 gene, which encodes glycoprotein receptors 3A. Our investigation identified that patients with heterozygote genotype have platelet dysfunction, which caused the development of the complications in heart failure patients. Another polymorphism was in UGT1A6 gene, which is responsible for the metabolism of the aspirin. Aspirin metabolites excretes faster with the presence of the mutant genotype of UGT1 gene, which suggested for heart failure patients to be prescribed with higher dosage of the aspirin. Our investigation concludes that these polymorphisms will help to prevent thrombosis and the bleeding events and to prescribe correct dosage of the anticoagulant treatment before and after LVAD implantations to improve heart failure patients' implantation time, which will improve their life and reduce mortality rate. Thank you.